Thank you, Rachel. Thank you very much for that lovely introduction. And welcome everyone to this evening's event. It's a real honour and pleasure to welcome so many of you tonight. It's been really interesting putting the presentation together. It was, say, a little bit challenging when I considered who was going to be in the audience. So I've tried to keep some of the science to a minimum. I know there's a bit of a mixed audience tonight. But what I wanted to do in the next 40 minutes or so is just take you through my academic journey and highlight some of my research highlights over the last 25, probably nearer 30 years now. So we will start where it all began, back in the 70s, where I grew up in a lovely town in North Lanarkshire called Airdrie. Now, I know some of you are very familiar with Airdrie. It's a lovely town. It wasn't uncommon to be walking down the street in those days and be handed a monkey. And this is probably where my love of animals first began. But I was born in 1972. I wasn't going to reveal my age, but it's out now. So 1972 to Tom and Phyllis. You can see the picture up here. I was a second child, born 3 years after Stephen. He's also known as the favourite. So he is here somewhere. Where are you? Oh, he's over here. My dad worked in the building trade. My mum, she had various jobs, from cook supervisor, manager in Pippity. And anyone of a certain vintage will recognise the pyjamas. Here, the Pippity pyjamas. But it was a loving environment, really supportive. Mum and Dad were certainly encouraged competitiveness with my brother and I. There was no gender inequality in those days either. We were both treated equally. And as you can see from my Scotland top here, I was quite a tomboy. So sport has always been important to me. So I went to my first school was St Dominic's primary school in Airdrie, and then went on to St Margaret's High School. It was quite a surprise to me looking back, like putting this thing together and reflecting on things. It was almost, it'll be 40 years this August since I went to St Margaret's. And this picture here is a 6-year common room with my two best friends, Jacqueline and Elaine, who are in the front row here. We've had loads of good experiences together, loads of memorable events in our life, and they've been very supportive, so thanks for coming tonight. But yeah, this is my first year picture. So I did science at school. I really enjoyed science. I did the 3 sciences, but my passion was really in art. And I did, I won the school prize, 4th, 5th and 6th year in art. And I wanted to go to art school, and that was my dream, was to go to art school. So all 6 year, prepared my portfolio, worked every day, was in the art studio, and submitted my portfolio to art school. But quite upsetting at the time, I didn't actually make it to art school. And on reflection, St Margaret's was perhaps not the most supportive environment for someone with those kind of dreams and ambitions. I think when the 6 years before I applied to art school, there was only one student who got to art school. He just so happened to be called Jim Dolan, but he wasn't a relative. But he was someone I really admired and looked up to, and that was the dream. We were all looking to get into art school, but anyway, never made it. Also at the time, we didn't realise at the time, but the actual equipment and facilities were pretty poor. You know, it was watercolours and pastels. There was no oil paints or acrylics. And when you looked at the competition at the time, you know, even there was a friend of mine at another school, she was painting in oils. And so at the time, I didn't really realise that was, I might differentiate my portfolio from someone else. It might not have. Perhaps I wasn't good enough. But I think coming from that kind of, I would say, deprived school, it didn't feel like that at the time, but perhaps not as, didn't have the facilities that other people might have, was a disadvantage. And it's something that's passionate when I'm looking at the kids and the pupils that come to this university. Is something that's really important to me, to give people from all backgrounds that opportunity to come to university. But anyway, it wasn't all bad because I got into Glasgow University instead. so as my fallback position, I applied to do biology and psychology. And it's around about this time where I met my husband, Andy. And it's probably just as well that I did put down the paintbrushes, because two artists in the one household might not have been a good idea. So Andy was he's been my supporter throughout over 30 years now. Andy's a musician and a writer, highly successful, but he's always been there to support me and encourage me all throughout my career. And if anyone's interested, oh no, wasted the surprise. The surprise was, if anyone's interested, his new book's out the 23rd of, his new book's out the 23rd of March, available at all good bookstores. So that was, sorry, I kind of wasted the punchline a wee bit. But. So I went to Glasgow University then. I did biology, psychology, and then I majored in developmental biology. And again, Glasgow University, met loads of good friends. Barbara came up from Brighton today. She's over here. I haven't seen Barbara for 20 years, so we've had a really good chat today. Barbara was my best friend at university. So it was in my fourth year doing my honours project with Tony Payne and Des Gilmore. I know some of you in the audience will know Tony and Des. That I really got the passion for science. I love being in the lab. Spent hours sectioning little bits of spinal cord, which is shown here, and counting neurons. Very dull, but I thought it was brilliant. I really enjoyed it. And that inspired me to go on and apply to do a PhD. And this is my graduation day with my mum and dad, 1993. So then on to do a PhD at Stirling University. This was under the supervision of Dr. Peter Kahuzak. So Peter was a really inspiring supervisor, worked really hard, and he spent hours and hours training me in electrophysiology, which is quite a technical method for measuring electrical activity in neurons. And this PhD lasted from 93 to 96. And again, I contacted Peter recently. He actually messaged me to say that he couldn't come tonight because he's actually working, he's a lecturer in Saudi Arabia now. And I told him, it was 30 years ago I started my PhD, and obviously that makes him feel particularly old because he was supervising me. But Peter was great, really inspiring. He'd come from Bristol University, really famous pharmacology lab there. But the project, I'll not get into a lot of detail, but it was looking at the effects of sensory deprivation on development of the brain. And it's a thing we call plasticity. So plasticity in the brain is the ability of the brain to modify and adapt its function in response to, it could be tissue damage or changes in sensory input. It's really important in learning and memory and how the brain repairs after damage. And it's also really important in pain, which would something I'll go into in a minute. But we use the whisker barrel cortex as a little model. The rat's whiskers are like our hands. They use the whiskers to feel their environment. And there's a large bit of the brain that's given over to processing information that comes in from the whiskers. So it's a really nice system to investigate or use to explore what's the consequences if there's damage to the sensory system. So we got 2 papers published from this work. It was really enjoyable time at Stirling. So then we're nearly going on to the vet school, but not quite. Yeah, so I put this picture up. So I did graduate in 1996. That's my mum and I, quite happy at graduation day. But I've got a picture here of my dad. So it was tinged by a little bit of mixed emotions, because my dad, he was killed in an accident at work the May before I finished my PhD. So I just put that up as a wee tribute to him. It was obviously a really difficult time for us all, but it almost helped me to concentrate and finish my PhD. I was determined to get it finished in the 3 years. So we all talk about resilience these days, but that certainly helped develop my resilience. So on to my first job at the vet school, and Andrea's sitting at the back here, so thanks for making it through tonight, Andrea. I remember when I first went from interview with Andrea, she was you were only Dr. Nolan at that point, you weren't quite Professor. And in the pharmacology department at Glasgow University Vet School, upstairs to the office for the interview, and there was these 2 wee black Labrador crosses, quite stinky, I must admit. And of course at the vet school everyone brought their pets to work. It was quite normal for folk to have dogs under the desk. So Molly and Finn came out to say hello, and I knew right away Andrea and I were going to get on well when I saw the dogs. So I was so lucky I got a job with Andrea. She's responsible for my introduction to the pain field, but also my love of sheep. So anyone who knows me knows that I've got a wee fondness for sheep. If you come over to my room, there's some things, sheep on the wall, and when I do my paintings and drawings, I quite often like a wee portrait of a sheep. So this was a brilliant time at the vet school. Andrea had secured a 3-year BBSRC grant. That's quite a prestigious funding body grant. It was for a postdoc and a technician. I was the postdoc. That's once you get your PhD, you get this postdoctoral position. And that's some of my colleagues at the vet school. It was a really, really happy time. I was there for 7 years. I can see Neil and Anna and Neil are here, and Peter, are you there? Yeah, Peter's over there with one of his kids. So the 3 of them are up the back row there. So we're still good friends. We do try and catch up now and again over the years. So that was 1996 when I joined Andrea at the vet school. And Andrea's been a great mentor and role model for me throughout my career. And we collaborated in research for at least 10 years. And I think I worked out we had about 15 or 16 publications and maybe about 30 conference presentations as well. So she worked me hard, I must say. She worked me hard. But I'll give you a little just kind of overview of some of the research now. So please bear with me if you're not a scientist. But the purpose of the research at the vet school was to improve pain management in animals. So to do this, we had to be able to measure the pain response in animals. And we use sheep as our model system. So sheep can develop a number of naturally occurring inflammatory pain conditions. So things like, there's a condition called foot rot, which is a bacterial infection of the hoof tissue, or mastitis, which is bacterial infection of the udder. So they were a great model for us. So we would go out to the farms and collect in the sheep and test to see how much, whether they were limping or how much pain they were in, and then give them various, not the normal treatment they would receive. We could try out perhaps some new drugs to see if we could reverse the pain and the hyperalgesia they were experiencing. So it was really, really great times. It was boiler suits, wellies, wading through shit. Sorry, am I allowed to cut that bit out of the presentation? The wee diagram here is just, I will be mentioning the spinal cord a few times tonight, and because the spinal cord is the area in the central nervous system that we were interested in. And the reason for that is because where the pain is first processed. So if you bang your finger or have a wound, you get sensory information travels up the nerve, and then the first place where it's processed is the spinal cord. And within that spinal cord region, there are changes that happen that cause a thing called pain, as we know, and hyperalgesia. Now, hyperalgesia, again, is a term that some of you might not be familiar with, and it's an increased sensitivity to pain response. And so in our animal models, we could assess whether animals develop hyperalgesia, whether they develop pain, and then give them some drugs to try and reverse that. So just very quickly about why are we interested in the treatment of pain? We've all tried painkillers. We all know whether they work or whether they don't. Some people respond well to painkillers, some people don't. And again, in preparation for this slide, I was looking to see what's anything new that's been developed, any new drugs that might be available for people to try. In a way, it's a wee bit disappointing that there hasn't been any magic bullet or any amazing breakthrough in pain research. A lot of the drugs that we're using certainly work for acute pain. So the three big ones are the opioids, non-steroidal anti-inflammatory drugs, which you'll all know about, is ibuprofen, aspirin, and paracetamol. These are still the three main drugs that are recommended for use. And if you look at the NICE guidelines, which is the National Institute for Healthcare Excellence, they actually, it doesn't matter what the condition is, it's those three classes of drugs that are still recommended. So despite all the work that's been going on over the last 50, 100 years, we're still really using the same types of drug. So why is it important then to try and develop a new class of drugs? Sorry, this is not very sensitive, my wee thing. So chronic pain is defined as pain that's been the individual's experienced for more than 3 months. So as I say, there's some of the drugs we use for acute pain, they work okay. Chronic pain is not very well treated. So chronic pain is estimated to affect 34% of the population. It increases as we get older, as I'm sure many in the audience again will testify to. It's unequal and unfair. So people in more deprived areas are more likely to suffer from chronic pain, and also people from ethnic minorities have a higher incidence of chronic pain as well. Now again, my other surprise that might have popped up here. Is this one. Okay. So this, I use a kind of slide like this for my students, and I usually get them to do a little vote. We've got wee buttons that they can vote. So I've got a question for the audience, and it's, which of these individuals do you think would be most likely to be the most sensitive to pain? The two variables here are women and men, okay? So we've got gender or sex, and we've got dark hair versus light hair. So does anyone want to shout out an answer? A. B. C. So there was some vocal people down the front thinking that the ginger man, well, he is quite sensitive to pain, but I don't think he's, so I'll give you the answer. So the answer is actually C. Oh, for goodness sake. I'm going to stop using my clicker. I'll just press my button. Yeah, the answer's C. So this is the lovely ginger lady here is my sister-in-law Maureen. And people with red hair are more sensitive to hot and cold pain. This is a little ginger mouse. Who would have thought you get ginger mice? But you do. So there's some studies carried out in the mouse, and they found there was a deletion in a gene known as the melanocortin-1 receptor gene. This is the gene that gives us their pigmentation and the red hair. So people who have ginger hair are more sensitive to pain. But also women, unfortunately, are more sensitive. And there's an increased incidence of chronic pain in women, and we see that right through the age range. From anything from back pain, headache, migraine, women are more affected by pain. So the answer is C. So sorry, Anita. So there's an unmet clinical need for chronic pain. Chronic pain affects the number of days at work. Some people lose their jobs. There's also comorbidity with suffering from depression. But in terms of my research, there's an unmet need in terms of the treatment for chronic pain. So 1 in 3 patients report their pain is not under control, and 1 in 4 patients believe their doctor doesn't know how to control their pain. And I think my mum was that correct. Yeah, I agree. So there's a need for new treatments. So I didn't even look to see what time I started speaking. So someone, where's Rachel, can give me a nod. Okay. Need for new treatments. So this is, again, looking at some of my research. So the purpose of the research is to try and identify a new target. That's what we call. So something that we can develop a new drug for that can block the target and prevent the pain. Okay, so probably over the last 25 years, that's really the focus of my research. So we've been looking for changes in gene expression in the spinal cord region in particular. So this is our spinal cord. This is a section through the spinal cord. And we used various animal models. That's one of my pictures of a sheep. I just put that in because they look quite nice. So just going back to then the vet school, just to summarise briefly some of the research findings. We did we managed to identify COX-2 we confirmed it as a target. There was just early work at that point developing COX-2 specific drugs. So these are special types of non-steroidal anti-inflammatory drug, that, things like diclofenac, rofecoxib is another one that are specific for COX-2. So we characterised when it was expressed, and after treatment we found that the COX-2 was down-regulated again. So this was quite exciting work. We then went on to look for another receptor called metabotropic glutamate receptor. Now you're not expected to remember the name, but we did loads of work on these receptors. There are 8 different receptors of this type. And I've just shown you a little snapshot of a couple of papers we published looking at one of these receptors. And this receptor was called mGlu-R5. Again, really pioneering novel work that we found that the mGlu-R5 is upregulated in spinal cord, so it was increased in spinal cord in response to inflammation, in response to pain. And then we went on to use new drugs that hadn't been tried in pain before. And again, we published 5 or 6 papers showing that if you modulated or inhibited those receptors, you could reverse the pain response. So these were really highly cited papers, probably some of my most highly cited papers, so I'm quite proud of that work. As I say, Andrea worked me quite hard at this time. But, the question then, though, and it's all, as a scientist and researcher, it's always about, well, so what? What's the benefit to man? What's the benefit to animals in veterinary medicine perspective? So we want to see, is there an impact? Is there translation of this work into a clinical development or a clinical drug? So we did identify changes in these receptors. That's key finding. The impact, new drugs have reached clinical trial. So that's really exciting. You don't need to read this. This is from a paper. This is a summary of some of the metabotropic glutamate receptor drugs that are currently in clinical trial. Many of them are in phase 1 and phase 2. So for those of you who don't know about clinical trial development, the drug, it can take from 10 to 20 years for a drug to go from an animal study right through the clinical trial phase before it can actually be given to a man. Or so there's first in man is phase 1 clinical trial before it can be used as a prescription drug, I should say. Okay, so it takes a long time. So these drugs that we studied probably 20 years ago now are still going through these early clinical trials. There aren't any available in terms of therapies. It's a wee bit disappointing, but as I say, a lot of these things do take a long time. And one of the reasons why a lot of them don't actually make it into a prescription drug, we'll call it, is because they have adverse side effects. So there was one developed for migraine pain that got to phase 2 clinical trial, but it was stopped because it caused liver toxicity. So it's very difficult to get a drug through those clinical trial phases because we're very cautious. It's all about patient safety now. And even something like paracetamol that we all use routinely wouldn't be available, or probably wouldn't pass clinical trial guidance nowadays. So now I'm going to move on to GCU. I joined Glasgow Caledonian University in 2004. And you can see here these two colleagues of mine, Professor Anne Graham and Dr. Tricia Martin, who's here. We started on the same day, the 1st of January 2004. So there's actually 5 of us started the same day, and we were employed. It was on the back of really successful research assessment exercise. I think I was trying to look back. I think it was actually 2001, the RAE. So this is the government do a kind of a review of all the research in universities, and then you get graded to see how good you are. And depending on how good you are, how successful you are, you get allocated funds. And it's an exercise that still goes on. It's very important to our university. So there was 5 of us employed at that time. And it's a delight actually to know that the 3 of us are still here 19 years later, 2 professors and 1 reader. So, you know, and it's and I think it's testament to the investment that was made at that time into early career researchers and young, talented, that was me included, young, talented researchers. You know, we have been successful. So here's some of my colleagues here from biological and biomedical sciences. You can see we're a shiny, happy bunch. A great department to work in. Colleagues are really supportive, collegiate, and bio is well known for liking a party or 2 as well. So I was there for a couple of years when I took on a programme lead role. So at this point, we only had undergraduate programmes in the department, apart from one. We had an MSc, which is a master's programme in biomedical science. That was for our hospital employees. And up until 2004, that was the only master's programme we had. So we had a big push to promote our discipline for a master's audience. We launched 3 new master's programmes, and we targeted the international market. And it's hard to believe back in 2004, the diversity of the student population was limited, shall we say. You know, there wasn't that many international students at all. But it was really successful. This is one of my favourite pictures and one of my best experiences. Anyone who knows me knows that I like to talk about India quite a lot. Does anyone know I've been to India? But that's me there getting blessed by an elephant. So that's and I think it worked. So I was speaking to Shiv about this. So Shiv lives quite close to this. There's a temple called Tirupati that I visited, and Shiv's familiar with it. So the wee elephant. Yeah, so this was a tour. We did a tour of Andhra Pradesh. We visited colleges and recruitment fairs, and it was a brilliant time. It was a time when academics and support staff, I think Claire's here as well. Claire, I think I saw you there, Claire? Ah, there you are. Claire and I went to India together. And, you know, it was a fantastic opportunity for staff to go and get a different culture and have a different experience. And then, of course, when the students came to GCU, we welcomed them, and it really added to the diversity and cultural awareness for the department. It was really enriched the environment. And we spent loads of time, you know, with the students. That's me and Jane Nally, another lecturer that worked with our master's students in the lab. And we went up Ben Ann one year as well with them, so it was really, really good fun. But so I think supporting the students is really key to the role I've had as well. Although I've shown you some of my research highlights, I think really, really passionate about supporting people, young and young scientists in their career development. And this slide is just some of my PhD students. Again, some of them are here tonight. Craig's here, and Nasser. Nasser's come all the way from Saudi Arabia, so thanks, Nasser. And they've all went on to have really successful careers in drug companies, associate lecturers, and associate professors. So I'm really proud of that achievement. So I'm going to, I'm not sure how long, Rachel, how long have we? 10 minutes, that's fine. So I just wanted to summarise some of the research that I've been doing most recently, and then I'll finish off with a couple of summary slides. In terms of my career, so after I became programme lead, I was programme lead for 8 years, and then I became assistant head of department for 4 years, then head of department for 4 years. But during that time, working with the PhD students and also some collaborations within the department, I managed to keep going with the research, and I moved into more looking at comorbidities with pain. So it's like comorbidities are conditions that come alongside the pain response. And one of the things that I was particularly interested in is obesity and diabetes and the link to pain. So individuals who are obese are more likely to develop chronic pain, and they've got an increased pain sensitivity. And my research then was looking at what is the cause of the increased pain with obesity. Now again, the reason for doing this is to try and identify markers or things that might change that cause the pain. And I'll show you some of the research we've got that's highlighting some of the things that we've found that change with obese individuals and obese rodents. But I just wanted to, I couldn't have a presentation without a picture of Bonnie. So Bonnie's our dog. Well, it's actually my mum's dog, but Andy and I share. Dogs share. We're thinking about what's the cause of obesity. So obesity it's not caused by one particular thing. It's not because people are greedy or overeat, although dogs sometimes do. But there's a really recent study that showed that Labradors have got a mutation in a gene called for the POMC gene, which means they overeat. They're 25% more likely to become obese. But Bonnie's not too bad. We need to keep a check on her. But it also means they're really easy to train as well. So that's why Labradors are really good guide dogs. And so I put her up because just to illustrate there's a genetic factor underlying obesity. Also some work we carried out with Professor John Kraft with obesity, showing that the microbiome changes as well in obese individuals. So the microbiome is the bacterial environment within our gut. And there's loads of recent work looking at the microbiome and how it might cause obesity. But the thing that I wanted to focus on for my research was looking at things called adipokines. Okay, so adipokines are molecules or proteins that are released from our adipose tissue. That's where the name adipokine comes from. And here we've got a little picture that shows our fat cells or our adipocytes. So when people get obese, the adipocytes increase in number and also they expand. They kind of blow up. And in obese individuals, they release inflammatory mediators. So they release molecules that cause inflammation. And molecules that are protective are reduced in obese individuals. And the hypothesis was for our work that this change in adipokine release causes the pain response. And that's what my PhD student, Tommaso, was working on, and Nasser as well. I'll show you some data for his work as well. I'm not going into a lot of detail in the graphs, but what we used was a model of rodent obesity. If you can see here, these rodents have a genetic mutation. It means they're hyperphagic. That means they just eat all the time, and they don't know when they're full, so they get obese. And the wee skinny ones, it's called a littermate. He's in the same cage as the fat rat, but he doesn't have the gene, or he does have the gene. He has the gene that's normal, and the fat one's got the knockout, okay? So the genetically obese rat, he develops more inflammatory pain, and he's more sensitive to pain responses, just a wee bit like obese humans get. And we also found then, if you look at expression of genes in the spinal cord, there's an increase in the level of one gene called TNF-alpha, which is a pro-inflammatory mediator, whereas the anti-inflammatory protective molecules were decreased. Okay, so keep that in mind. Similarly, in diabetes, so we could develop a model of diabetes. And this was work with Professor Anne Graham in Nowara, another PhD student. And similarly, what we could do is measure the pain response in an animal with diabetes. The pink line shows a normal animal, and the kind of blue line shows the diabetic animal. And over a period of 14 weeks, the animal with diabetes develops a sensitivity to pain. So again, this was a useful model for us to investigate what's the cause of that increased pain. And some of our publications then went on to show, again, there was what we call a dysregulation in adipokine levels. So the top graph shows we took blood samples and we measured these adipokine levels within the blood sample, and we found that there was an increase in the expression in some of these adipokines, which are pro-inflammatory, so they cause inflammation, and some of the anti-inflammatory ones, which are protective, were down regulated or reduced in levels. So therefore, the hypothesis is, okay, so this change in adipokine expression, does that underlie the pain response? And if it does, if we block the adipokines, so we get a new drug that targets these molecules, can it reverse the pain response? And that's what we then went on to show. So worked with from NASA and also Tommaso, they used two different blockers. One was a blocker, so we used visfatin, which was an anti-inflammatory marker, and then we used another drug that modified adiponectin expression. Just to summarise, I know it's probably it's quite complex to get into the details, but by modifying the adipokine level, we were able to reverse the pain response, and that's really the take-home message here. So it's just really showing that there's another marker or something else that might be a target for the development of a new drug. My thing's playing up. Don't look. I'm putting this down. So just the final few slides then. I know I had plants in the title, so I thought I would just give you a quiz at the end. So some of the work, my very first PhD student, when I came here, I was still collaborating with Andrea Nolan. We had another grant about metabotropic receptors, but we started to move in to look at medicinal herbs and whether they might be therapeutic targets for pain. And I put this slide up. This is one I show my students when I'm teaching about pain. And I usually get them to shout out the answer to what they all are. So I don't know whether you want to do that. I don't think I've got time. So I'm sure you pretty much recognise most of them. Of course, this is the most famous one, Andy Gardner's favourite flower. That's the poppy. So we get opioids from the juice of the poppy. I've also got cannabis. I want to shout out whose favourite that is. This one here is the cannabis. Magic mushrooms, I'm sure you know. Does anyone know what this is? Coffee. Yeah, well done, coffee. So coffee is sometimes not regarded as a drug as such, but it is it's a drug. So anyway, this one. So just this one here is. Anyone know what this is? Ginkgo biloba. Yeah. So Ginkgo biloba. Well done. So this again, I'll not get into a lot of detail in the terms of explaining the graphs, but Laura did some work. We got an extract from a company in Germany called Schwabe Pharmaceuticals. So they extract the alkaloids from plants, and they make a standardised extract, so you know exactly what's in the chemical that you're getting. So it's not like you're mashing up plants and using them. So this was really a good collaboration. They gave us some money subsequently to do some more research. But we found that Ginkgo biloba could reverse inflammatory pain in a dose-dependent way. In fact, I think Alistair Corbett was on this paper. Where are you, Alistair? There he is. I think your name's on one of those. I'm sure it is. And then again, we used the extract to see if we could block some of the inflammatory mediators. So we found it could inhibit cyclooxygenase 2, TNF-alpha, and inducible nitric oxide synthase. So that was quite nice. So I did move away a little bit from the medicinal plants for a while to concentrate on obesity research. But subsequently I've kind of moved back to looking at medicinal plants as novel therapies for pain and analgesia. And Stephen and I have got this is Stephen here, Dr. Patterson, a collaboration with Chinmay, his PhD student, looking at Indian natural products, so Coctus pictus and Japanicides. And we've found that they have anti-inflammatory and anti-obesity properties. So some of that work's Stephen's published with Chinmay, and we've got a little paper in preparation. And then my new student who just started in November, Fatima, I think Fatima's at the back there. Hey, Fatima. We got a Commonwealth Scholarship, so this is a prestigious award. So Fatima joined us in November from Nigeria, and she's brought with her some Nigerian plants called Parquetina nigricens. So we've just started that work, and it's looking again, can it block insulin release? Are they going to be anti-inflammatory, these products? So Fatima's extracted the alkaloids from the plant so far, and we're just starting that work. So it's really, really exciting, really interesting. So that kind of brings me to the end of the research. I'm sure you'll be glad to know. Just to summarise, I apologise for the emojis because I know they're a bit naff. But just to summarise some of the work and the highlights then over the past 25 years or so. We have identified new targets for the treatment of pain, whether it's metabotropic glutamate receptors, adipokines, or whether it's medicinal plants. And then we went on to look at some new drugs, whether we can modify the expression of these things. And in rodent models, or what we call preclinical models, we certainly got some positive results. The drugs do work, as I've said in the title. The next step, though, is the new therapies. How do we translate the basic science, the preclinical work, into an actual therapy that will benefit us and benefit man, and also animals as well, and we think about veterinary medicine. And there is there is promise, there is some new drugs in development, but as I said earlier, it takes a long time to get through that clinical phase and into a prescribed drug. And I think most of the new drugs that are available are still based on revised formulations of the opioids or non-steroidal anti-inflammatory drugs. We haven't found the magic bullet yet for pain, but I do have promise that we will make a breakthrough. So just then to finish up and summarise, just to say really thanks and acknowledge some of the people I've worked with over the years. I've got a wee list at the top here. It's previous and current heads of department that I've worked with. I met Les Wood on the way out tonight. I was the first one here's Les, so he was my head of department, and then Alistair, and then Anita took over from Alistair, and then it was me, and then Linda, Professor Scobie, who came from the vet school as well, is now head of department in biological and biomedical sciences. So it's a real privilege to be the head of department of bio. So that was a great experience. So now I'm moved to being associate dean for research, and I'm just meeting loads of new colleagues, which I'm really enjoying the experience. I've only been in the role 6 weeks, but I'm thoroughly enjoying it. I do miss bio a wee bit. But it's really important, the role models and the mentors that I've had, I probably wouldn't be here without those individuals guiding me and helping me along the way. But just, it's really important for me also to make sure that I can mentor young people coming through as well, and in particular, but not only young females, but particularly young females, because we know in science that females are underrepresented, and in particular when it comes to leadership positions, females don't progress. In research in particular, they don't progress through that career pipeline. So that's one of my kind of goals, as well as being Associate Dean of Research, is to support the young women coming through. And I thought this picture was really nice. It's the grad ball last year, and that was my 4 honours students. So honours students, they're the students that are doing their final year project in the lab with you. And they used to come in with their hoodies and their leggings on, and then, you saw them at the grad ball, and they just look absolutely lovely. That's me, I'm in the middle there. So that's really important. And then finally, the team. I can't leave without saying thanks to the team. Again, without the team effort, I wouldn't be in the position I'm in either. It was so being a this one in particular. So my team is the bio team. I've got a new research team right across the School of Health and Life Sciences, and I've also got my senior management team. We went to walk at Christmas time down to George Square. I never made it. So just to say thanks to everyone that I've worked with, all my colleagues present, but also past as well. Really do appreciate it. So I've got a few acknowledgements. People I've worked with, there's loads of co-workers and collaborators, both past and present, people that I've published with, and my PhD students, and also some of the sponsorship I've had in along the way. But of course I can't leave without saying thanks to the most important people, and that's Andy, my husband, so thanks for all your support, and my mum sitting here too. So without her guidance and tell me, have pride in yourself and have the confidence to try new things, I wouldn't be here. So thanks to you and you. Now, I should say I don't want to blow my own trumpet, but I've been doing it for the past 45 minutes. Just to let you know, I still do a wee bit of painting. And you can see that I quite like animals. Yeah, and if anyone wants a commission, no.
If you want to take a seat, you're happy to stand if you want to. Well done, Sharron. It was a fantastic and inspirational lecture, and a real talent to present it to a non-expert audience, but also, I think, shining a light on the importance of team science and collaboration, which really was inspirational for me. We've got about 10 minutes for some questions, if you've got any. I've got 2 hands up already. Carol, Professor Emsley, please, go ahead. I'll come and sit with you.
Really engaging, so always great to put this together. I was wondering, apart from being blessed by an elephant, what advice would you have for people starting off their academic careers, particularly in your discipline?
I think what Rachel said is really important is the team element is key. I think you can't be, nowadays in particular, you can't be doing research in isolation. I think it's really important to have those key partners that you can rely on. And ask for help as well, and seek out, new expertise that can feed into your project.
There was, I think, another question about halfway up. I'm going to have to stand to see, and then I'll come to Andrea, Professor Nelson. Go ahead, please.
You spoke about the reduction of adipokines in the body. Was that so through the drug that was being used at the time to do that? Is that like a gradual reduction or a permanent reduction? Is that like a long-term solution, as we were speaking of chronic pain in that case, or is it more like you have to continue the drug like for the rest of your life to be able to sustain that kind of lifestyle with lower pain?
So again, it's a good question. The models that we use are short-term, and I think because it's animal models, ethically we're not really able to do particularly long-term studies. So it's a difficult question to answer. I think knowing the way that the body functions, it would probably be a long-term treatment. It would be a management option rather than a reversal. But that work hasn't been done yet. So,
Andrea, please. I'm going to have to stand up. I'm too short to see.
Thank you, Sharron. That was really fantastic to get that whole picture. What I was really struck by was actually these research careers take a long time, and bits of work take such a long time, don't they? You're talking about this sense of disappointment that like some of these target drugs aren't ready yet. But I was interested in how you keep going whenever actually when you're in your research training, you're thinking of projects that take months and months, and then as you get on in your career, you start doing programs of work that take years and decades. So how do you sort of keep going, knowing that actually it's a very long-term journey and research question you're on, not just one of those quick fix projects, go in, analyse something and come out?
Yeah, I think you can probably see from the way the research has changed, looking at for different targets. So we started off at finding about COX-2 and INOS, then metabotropics. We have moved around a little bit. So throughout the research that we've published, we're finding new targets. But I know what you're saying is, what's the motivation for keeping going when they don't translate? And I think as a basic scientist, it's not the impact in the translation is probably something that we didn't think about as much as we do now, and I think it's been really useful going through the REF exercise and being part of the School of Health and Life Sciences. Whereas as a preclinical scientist and a basic scientist, we're now focusing much more on what's the impact of our work. So even although the data I've shown you hasn't translated, it doesn't mean it's not informing drug companies. And as you can see from the list I showed from the metabotropic glutamate 5, there's loads of companies are working on that, even though it's not, what's the word? It's maybe not, the link is not obvious, but I think the work that you do as a preclinical scientist is really important to inform those clinical trials. I don't know if I've answered your question, but...
Any other questions in the room? Yes, go ahead, thanks.
Sharron, a great story about the background in Airdrie and St Margaret's, etc. Obviously, as a former pupil there, I can understand it. You've been to India. Have you ever been back to Airdrie and back to your school and told this inspirational story back to young women who are maybe studying science at school?
No, I've never been back to St Margaret's, but I would be happy to go back. We do a lot of community engagement and outreach. The bio team up the back there, a lot of them, we do go to various schools, and it's something that we're probably be looking to increase. But I think going back to your own school would probably be beneficial. I'm sure I could. I know some people that work there, so.
Any other? Yes, right at the back there, please.
Sharron,
Uh-oh.
you were very coy, perhaps, about your biggest achievement as head of department, and that was getting the second floor corridor painted. So I now appreciate quite why it was such a passion for you, and I don't understand why you actually didn't do it yourself. Are they blue or purple?
They were supposed to be blue, but they are purple.
This goes back to the fact that Sharron did the bad paints when she was a kid.
And I'm colourblind.
Something about why she never get into art.
Possibly.
We've maybe got time for one more. No? I think it might be time for refreshments in that case. Can I just say what...